brownish sponge, and the thought is that the sponge is no longer alive because it is turned brown with sediments on it, and maybe even some precipitation of the iron oxides that are in the water here. Typically those sponges are brighter white. So maybe a little, pilot a little to the right of the sponge so that that end is intact if we can get it.
Which end?
The end forward, closer to us. If we can get that intact, that'd be great.
Okay.
So again, these red dots, the laser dots, are 10 centimeters across. So this is a small rock. Hopefully we can get just, oh yeah, oh.
Nope.
Well, whatever you get. Oh,
that is quite brittle.
It is very brittle. It's just going to disintegrate in the box, isn't it?
I had it on a very low force as well.
But it would, yeah. Okay, well, maybe we'll find something else here. We've certainly looked at these.
Was the sponge the priority or the rock?
The rock. The rock was. Yeah.
Okay.
I mean, if you want to try one more time, that'd be great.
So you just want a chunk of it?
Yeah, I'm afraid it might disintegrate in the box.
We might have to put it in a bio box if we.
No, we don't want to use a bio box for this then. So.
I would imagine this is probably going to disintegrate in the rock box. I think the only way to make sure it gets to the surface is to put it in the bio box, which we can do.
No, I don't want to do the bio box.
Okay.
We'll have plenty of organisms. Can we just do one last zoom on that fresh surface that just broke, because that will tell us a lot about what the rock composition is.
I'll go ahead and do another squeeze too on this upper part.
Okay.
Once we get a zoom. All right, video. There, hold it there.
And what a lovely squat lobster is on the camera now.
I'm amazed he's still there after all this trauma.
He's going down with the ship.
He's daring us to take his home.
I'm going to zoom out a bit, video, and then I'll give it a squeeze here.
So you can see the broken away surface is the color of the surrounding sediment. So this is just slightly consolidated sediment.
Yeah.
And the surface of it that makes it look like a solid rock is the precipitation of the iron oxides. Okay.
All right.
We tried. We got some good video.
We'll keep looking for you.
And we proved that it is not a very solid rock.
Thank you.
Thank you.
We could move the mini Zeus up, co-pilot.
Coming up. Does that lid close itself? We'll have to see. Last time it didn't.
Oh, it doesn't?
Well, you want me to check real quick?
Yeah, go for it. I assumed it does, right?
That's what I assumed last time, and I had one time that I got stuck, like that.
Oh.
Yeah.
Oh, I wonder if it's the new springs.
I'm going to come back inboard for you.
Okay. Thanks.
Just wants to be open.
Okay. If you could bring the mini Zeus over.
Okay.
Great. Ooh, it's stuck on something.
Come down. Am I all the way? Yeah, I'm all the way out. Let's see. Can we see if it's? You want me to try to move the other arm out? It's stuck underneath there.
Should be all the way outboard, right?
Should be.
It's stuck on the base.
Oh.
I'm torquing it.
Draw out a little bit.
Yeah, draw out, and that'll get it off the base of the shilling.
Coming out. Okay, there he goes.
There you go. I think it's because
now you got the white. Hold on, let me get a little further. There you go. That should do it.
All right. Jeez. Those springs are awfully tight. See how it was like more than halfway and it was still pulling back?
Okay, I'm going to come back in.
All right. I'll take Mini Zeus and bring it back over.
Okay, great.
Maybe we should move this, you know, those holes at the springs mount to?
Yeah.
We should bring it up. Yeah, bring it up a bit and loosen those a bit.
Okay, we're at a depth of 1,690 meters. We just attempted to collect a rock sample, but it turned out to be too brittle. So we at least exposed the inside of the rock, and that tells us a lot about the iron that's been precipitated out of the water column in this area, and that's why you see that brown layer. But once it's exposed for a long time, you don't see that brown layer anymore.
Indexed out.
Hydraulic secure.
And we are also very close to the escarpment now. This is the base of the escarpment, and we're going to be moving closer to the wall.
Sure. Okay. So what's our direction of motion here, Nev?
310.
310, so we'll have to bring Sirius over a bit.
305, somewhere in there.
Okay. I'm going to come up and saddle to starboard.
You can see that there are several corals and or sponges on the horizon here.
Seems about right.
Yeah, 305, 310, somewhere in there. Yeah. I'm going to saddle over and you can turn your head a bit.
2315?
310.
310.
Yeah. And I think we could go ahead and get a move. Looks like there's a lot of coral up there.
Meters, 310. Bridge, this is ROV nav.
This is Bridge, over.
Yeah, we'd like a ship move, 30 meters, bearing 310 degrees, speed decimal 2 knots.
ROV nav Bridge, copy, 30 meters. 310.
Looks like we have a few sponges coming up here.
0.2 knots.
Good copy. Thanks, Bridge. You got Levi down here now, by the way.
I think I can get up there. Take a look.
Also looks like there's a tube anemone lower left. The maroon colored. Wow, look at all these sponges. That's amazing. So in general, we have learned a lot more about corals and octocorals, scleractinian corals in this area compared to sponges. There's a lot of diversity of sponges, and they remain poorly studied so far. This looks like a great area for
ROV nav,
a variety
Bridge,
of sponge
initiated.
species.
Over.
Move initiated. Good copy. Thanks, Bridge.
And hopefully our sponge expert Shirley Pomponi will join in and tell us a little bit about some of these sponge species. Oh, but it sounds like she had to leave. Oh, bad timing.
Yeah, I was referring to Joanna, our other sponge expert. She was in the chat room a short while ago. I don't think Shirley's been on yet today.
Well, that's too bad. Hopefully she'll be able to look at the video later. it looks like a chrysogorgid in the lower right octocoral.
More like chrysogorgid.
Would you like a closer look at that? Sure. Sure, absolutely. And you're moving toward me.
I am.
Okay.
Yep. Ship started to move here. Does the heading from Sirius come from USBL? Or?
You're talking about in nav? No, it actually comes from
the compass, right?
Compass. We fixed it.
Yeah.
Oh, in the high pack, yeah.
That might not be the right drawing for it, though.
I agree with you, Cheryl. This does look like a chrysogorgia, and this is a member.
Good, zoom in video.
The name chrysogorgia is a genus name. It's one of the octocorals in the family Chrysogorgiidae, which means golden corals, which refers to the sheen of the skeleton, which is kind of below the tissue. But you can kind of see that it's appearing somewhat golden. And they have very particular geometric patterns to the branching, and it's those geometric patterns that are used to identify the different species. And I couldn't tell you right now what this species is. But we're also looking at a shrimp that's in the branches, and we very often see these shrimp living with the Chrysogorgia. There's probably a very close relationship of a particular species of shrimp with a particular species of Chrysogorgia. And you note that bright purple color. I believe those are eggs that the female is carrying on the swimming legs. And there might also, I see there's something that looks like it's curled around the branch, a little sort of pinkish orange thing, and I can't tell right now.
Looks like an amphipod, possibly.
Is it a polychaete, maybe?
It could be a polychaete or an amphipod, I imagine.
Yeah, I can't quite tell.
It's a beautiful view of that shrimp.
Yeah. Feeding on something.
Yeah, through the branches of the coral.
I guess the purple is internal, isn't it? So it's not eggs. So it may be that she's developing eggs that'll be released later. So I wonder if that's a reflection of the internal gametes.
It looks like there's purple eggs on the underside as well.
Yeah, I think it looks like there may be eggs as well. Beautiful coloration.
Yeah.
And really nice shot of the coral, too, with that golden color.
A little bit. I'm just...
Yeah, it also looks like the polyps that you see look like they're kind of swollen. The polyps, I mean, each of the structures that's arising from that coral has a ring of tentacles and then a small body below that ring of tentacles, and the body is on the skeleton itself. And it looks like the bases are somewhat swollen. They're kind of bright white.
Good enough view,
and that would suggest that all those polyps are also undergoing a reproductive phase and are just packed with gametes.
Wow. And this kind of information is really important because every species has a different pattern of reproduction and when they spawn, and so being able to actually visually see that there could be eggs in this coral is information about when they may spawn.
All right, is that good for your purposes, watch lead, or do you want to stay here a little longer? Okay, let's go ahead and zoom out. Thanks, video. That was a nice shot.
We have some more of the pancake urchins. Looks like 3 of them in the view now. 1 close and 2 off to the right. And lots of exposed rock here.
This is pretty late right now.
ROV nav, move complete.
But this seems to be an area that's dominated by sponges instead of corals.
Okay, pushing ahead.
And their sponge habitat is also important, creating 3-dimensional structure on the seafloor.
Delta.
And they're also very prone to human activities such as trawling. If a trawl net came over this, it would probably
zoom in past the skids,
just
Bob.
take out a lot of these sponges.
Feel free to do so
they're
anytime.
also vulnerable species.
Coming down.
Nice view of that transition from the rock to the sediment in Sirius.
Yeah, if anybody wants to switch to live stream 2, and you can see the Sirius view of the D2 looking at the rock features, it's really a great one. A nice view to get an idea of the layout of this kind of high area that we're at right now. If anybody has a particular sponge they'd like to stop and take a closer look at, it looks like a lot of them might be similar species here.
Looks like we've got some larger ones coming up.
Oh yeah, we have some nice ones coming up on the right side. Some larger sponges.
Copy. What do you see?
Crinoid bottom screen going off screen right now.
Any interest in the crinoid at the bottom of the screen?
Sure.
Okay.
There's a crinoid that we're going to try to get a closer look at.
What time did that move in now?
Oh, there it is, stocked crinoid.
Yeah, stocked crinoid.
Right close to the lasers now. Oh, and there's a can.
Marine debris.
More marine debris to the lower left.
Oh, yeah.
It's kind of amazing to think we are so far off the shore that we would see any signs of humans, but unfortunately we do almost everywhere we go.
We don't have too long here. I think we're just about done with our move, but I'm still moving.
I think we're okay. It's pretty flat, and
okay.
I mean, you should be settled up pretty soon.
Okay, I'm maxed out. If you go off here, I won't have you.
That's okay.
Oh, that's beautiful.
Wow.
Said it ended like 10 minutes ago.
2 minutes ago.
2 minutes ago. Oh, yeah. Yeah, I think we'll be all right.
We got, what, 4 minutes or so?
Beautiful.
Ooh, up. Missed it. Can we come out a little bit? Yeah, hold it right there. We can get him curling his arms back.
So this is a stocked crinoid, so it's an echinoderm, therefore related to sea stars and brittle stars. And this one is on a stalk in order to get the main body, the main feeding structures, which you can see it kind of folding and unfolding or wrapping around and then letting out. They capture particles in the water column. In order to be in the water column, that's why they're on the end of this long stalk.
Very active.
You can see right in the center there, there's sort of 5 branches, 5 sections, and then those branch off into 2. So again, that's how you can tell that it's an echinoderm, because they usually have pentaradial symmetry, or multiples of 5. Which is, if you were tuned in earlier, we were talking about, that's not always the case, because in the case of sea stars and brittle stars especially, they can lose their arms and regrow them. So sometimes you'll find sea stars that just have 4 arms.
Thanks, Bob. Looks good. Sure. This one, right center screen. Okay.
This sponge here, isn't there a hedgehog sponge? Anyway, it kind of looks like a hedgehog with all those little spines sticking out.
Yeah, that's Polypsenia. We saw one of these earlier.
Okay.
But we haven't had a good look at these very large sponges yet that are kind of like radar dish shaped. There's several of them. Maybe if we can get a close-up of one as we pass by.
Yeah, that'll be great. I'll ask for that next. Oh, it's under another stock crinoid just to the left as well. Wow, that's beautiful. And that looks like a little tunicate on the rock wall to the left of the screen.
And just below that tunicate looks like a pteropod shell, kind of just behind part of the sponge. So again, pteropods are snails that live in the water column, but when the animal dies, the shell sinks down to the bottom. So throughout the course of this dive so far today, we've seen quite a few different species of pteropod shells.
Okay. Do you want to go ahead? Sure. Sure. Is there anything else on the screen you want to look at before we go over to the other sponges? Okay, let's zoom out. Let's come full wide. You can tilt the camera down a little more, Andy. Get a few more degrees.
Can you find them in the Titan, I wonder?
Yeah, that'd be helpful.
I can spin around. That's what I'm going to do.
That might take all the cable he needs, though.
Or should put me behind him. And
We're going to take a closer look at these sponges.
We're just going to look at this and move ahead.
Seem to be the dominant sponge type on this feature here. We are at 1,685 meters depth.
Does that look good, Andy? You happy?
I mean, there's no change.
Okay. All right, we can get lasers off. And then go ahead and zoom in a bit. And hold there, maybe. Okay, you can go in tighter. Okay.
So these look like large glass sponges, hexactinellida, and I'm not a sponge expert. I'm just kind of looking at some of the previous images, especially we have a lot of sponge images from the expeditions in the Pacific, and this looks like sponges from the family Pheronematidae.
Is there anything in particular you want to look at here, watch lead, or
a good start?
Did you get what you need? Oh, there's a huge.
You can see they obviously need to attach to a hard substrate. You don't see any of them growing in the sediments there, but where the rock outcrop
did you get what you need from the sponge?
Is showing, they're doing quite successfully. And these are also suspension feeders, so they are running water. They basically have a kind of a plumbing system. Water moves through the tissue, and they can remove very small particulates from the water column, probably stuff the size of bacteria, maybe organic particles that individual cells will feed on.
We'll see what we can do here.
Great, thank you.
While he's doing that, do you want to do a quick zoom video out there?
Not more. Ooh.
Looks like a giant.
Wow, look at that Brisingid sea star.
Oh, it's a sea star. It's massive.
Yeah. Lasers. And another glass sponge.
Yeah.
Yeah,
oh, that's multiple different kinds of sponges in a row there.
Yeah, the 3 different types we've been seeing all stacked up next to each other.
That's huge.
Yes, and then a Brisingid sea star that's bigger than any of the sponges, it looks like.
Okay, let's come out video. All right, Andy, looks like...
okay, fine, yeah, I want to go back to...
Let's come out full wide. Do we want to go see that sea star? watch lead? Sure. Okay. Okay.
There you go.
All right. Do you want to go ahead and put a short move in?
And while we're
in that direction,
moving the ROV a little bit to get a look at that sea star, the Persingid, I wanted to pass on something that Jason Chater from USGS and Woods Hole is telling us about
maybe
the
15.
rocks. He said that his work has been concentrated on to the south of here, but that these rocks actually may be a relatively thin crust of dense carbonate with minor amounts of quartz. So, in other words, not a framework type of carbonate.
So if you just move
the color is,
Sirius forward,
as always,
I think we'll be all right.
iron manganese, which can be quite thick out here and even forms tabular slabs.
I'm at 95.
Yeah, thanks, Jason. I just got back from lunch and I see that. Yeah, this region, these tabular slabs of manganese iron, these are precipitates.
Bridge, this is ROV Nav.
This is Bridge.
The Charleston Bump, which
like
is west of here, has also
meters,
phosphate,
bearing
and they can be
0 degrees, speed decimal
become
2.
extremely solid surfaces.
Understand, range is
Jason, if you're still
meters,
in the chat room, I think you are. Maybe do you think these would be worth trying to get a sample of, or do you think they would be similar to what we tried to collect downslope?
We also have some potential IDs coming in about the sponges. Afrocalistes is one, and Faecalia is another one. I think it was the middle sponge that we were looking at just a little while ago that might be the Afrocalistes.
Yeah, the Afrocalistes would be the one that had the little out pockets, look like little fingers arising from it around kind of the vase.
Great, and that's a glass sponge, correct?
Move initiated, thanks, Bridge.
Yes, that's correct.
And here we're getting our first view of the other side of the Faecalia, I believe.
Are these nicknamed amphitheater sponges?
Just watch the tether there, Andy.
They should be.
A little bit awkward, but we'll get there.
It's coming around.
You can really see how much of this rock has that iron manganese coating on it.
It's a little over you, but it's getting close.
Yeah. Yeah, it's just a little awkward. That's all right.
You can see that these have a lot of surface area, so they're able to catch particles in a larger part of the water column.
Be advised. Okay. I got the move going 090 and Cirrus heading 075.
Okay. Maybe I'll go ahead and come up a bit and turn. It's kind of an awkward angle there, watch lead. Did you need more of that, or?
No, I think that was helpful. Thank you.
Okay. So as you move closer, you can definitely come up in altitude, Andy.
Copy that. Sitting at 15, coming up.
Is there any, do you think we should collect one of the sponges? Or?
I want to go see that sea star. It's huge.
It's the biggest Persingid I think I've ever seen.
We
It's getting pretty hot video. Do they, these sea stars usually come in at this size? Is this something that we usually see? Oh. Okay, you want to get lasers off and we'll do a quick zoom?
Lasers coming up.
Go ahead and zoom in video. Hold there.
That's beautiful.
Has anyone mentioned that this is a Bersingid asteroid?
We did on an earlier specimen.
I think we did, but we were further away, so maybe it is a good time to repeat that. Bersingid sea star.
Yeah, this is a sea star that is a little bit different from your typical sea star, or what most people might be familiar with as sea stars, because it curls its arms up off the bottom and feeds out of the water column instead of on prey that are on the bottom or in the bottom. And you can see those long spines that are
was there talk of
arms.
Collecting a sponge?
Each of those spines is covered with small hook-like structures that help capture material from the water column, and then it can use the tube beak to transfer all that material down to the mouth, which is underneath the central disc facing the bottom.
All right, that move should be getting close to complete.
Cool.
Andy?
Great. Shipwise or me?
Shipwise.
Looks like I have about a 4-minute layback.
Yeah. I got uphill at maybe 310. I think we want to do one more. After this, we want to do like one more hop uphill.
Let's come out video.
Not if he's going to collect.
After. And then we can take a right.
So this whiter sponge here, the one we're right on top of.
ROV nav bridge, we are at our new location.
Good gap bridge, ship move complete.
Hang on just a second, Cheryl, for that question that you just posed.
Okay, we're debating whether this would be a sponge that we might want to collect.
Okay.
It's in lower abundance than the larger sponges, and so we're going to just kind of get an idea from the science team on shore which one would be more beneficial to the science needs.
Sure. There's also some sponges further to my right that look like they have a different shape. We can peek over their video.
Or a couple more hedgehog-looking ones, maybe.
Or garbage out in the distance. Yep.
Yeah, like another aluminum can or something. Looks like in the distance.
Oh yeah, at the top of the screen. More marine debris. And it looks like a royal red shrimp on the bottom part of the screen.
So any word on the sampling, or if not, we might go ahead and put a move in uphill and continue on our track. Sure. Sure.
Yeah, so Dan, the waypoint's got us maybe 35 meters uphill.
Okay.
And then taking a right and following that contour.
Oh, really? That's the plan anyway?
Yeah.
I mean, this almost looks more interesting. This looks like maybe the contour they want to follow.
That's what I was thinking, is maybe this. There's quite a bit of stuff here.
So it looks like just sediment up there. And this is the transition, right? So
I'm wondering that too, if we just want to follow this.
I think I see a Paramaricia sea fan there. Is that the first of the dive?
Zoom in on that.
Yes, I think so, Scott. And it has its commensal ophiuroid.
Yes, very characteristic for this species of sea fan to have one of these ophiuroid brittle stars in its branches. I'm not sure. I guess it's just growing from the rock behind the sponge, isn't it? Where I'm sitting, it looks like the polyps aren't fully extended. I can't see the tentacles on all of them, but they can withdraw completely into that yellow tissue to protect themselves, presumably.
Okay, you can go tighter.
Beautiful video.
So that's
down
just
to the
one
disc.
brittle star, isn't it? Or is it a sea star? It's a sea star.
You were right the first time. It's a brittle star.
It is a brittle star, okay.
Ophiuroidea.
But it's just one.
It can both wind its arms around the branches and basically overlay them on branches, so they're not necessarily always completely wound around. Sometimes they're just kind of draped back and forth, which allows it to more easily extend the arms. And the presumption, I believe, we might hear from either Tim Shank or Les Watling more on this, but the presumption is that this brittle star is feeding on things that land on that coral. And so, in a sense, maybe cleaning that coral off or protecting it from other stuff overgrowing it, as opposed to feeding directly on the coral.
Watch this is ROV Nav.
Go ahead, ROV Nav.
Yeah. The dive track has us going uphill another 50 meters and then taking a right and going over to waypoint 3. However, would you like us to follow this contour? It seems to have quite a bit of life here.
Can maybe mention it looks very sedimented up ahead.
Yeah, if we go towards the waypoint, I'm worried we're going to get into a sediment field, which may be...
So we're looking upslope?
Yeah, this is upslope. You can see the transition.
Do we have the lasers on those sponges just to get a sense of the size? I thought they were quite large, but now I'm seeing that there's a big asteroid next to them. Maybe they're not as big as I thought they were.
More directly uphill.
Or that's a very large
sediment. Or B, we follow this trail, which is towards waypoint 3.
Lasers are on.
Okay, understood.
I'm going to turn to center up you and the ledge.
Yeah, my pilot can take a peek and see what he can see in the distance, but I suspect this is it. But yeah, we can do it.
Well, first, any word on the sponge? Are we going to be collecting this, or should we move on?
Yeah, watch. My pilot's wondering if we want to collect or not.
Scott, is this something that you think we should collect, one of these sponges, before we move on?
Yeah, I still haven't seen good consensus. So that bright white one, if it's Aphrocallistes, then that is something that is of interest. But I don't know that we have the appropriate people to absolutely identify it. So I guess we should just move on.
Okay.
All right.
So we're going to take a peek up slope. I'll just push out at a tether length, and we'll see what we find up there, and then we'll make a decision about where to proceed. So Andy, you can follow me over and give me a little bit of tether by coming down.
In the meantime, Eric Cortez from Temple University added something to the chat room that the brittle stars after the Deepwater Horizon Gulf, in the Gulf of Mexico after the Deepwater Horizon instrument actually cleared off the Paramaricia. And the corals with brittle stars actually did significantly better in recovery. That's very interesting.
So I'm just going to push out, Andy, and you can come down to give me more tether.
Will do.
Go ahead and tilt your camera up.
I'm working on it.
Okay. Okay.
Okay, watch lead, this is now.
You want to bring out the background, Bob, as much as you can? We're just trying to see as far as we can in the distance. Doesn't look like there's much.
Hey, watch lead, this is now. So yeah, this is where the dive track kind of has us, kind of in the sedimented area.
Okay.
So this would be where we would turn right and follow the contour. The other option is kind of going back down where we were, where those sponges were, and then following the trail of sponges towards waypoint 3. So there's two options. Either. Okay. Understood.
Okay.
Yeah, we thought so too.
Great. We'll come back.
Thank you, Watson.
Okay, so we're trying to make a decision about where to go next. It looks like if we keep on this heading, we're going to be traversing over more soft sediment. So we're thinking that we're going to instead follow the rocky ledge and the sponge garden to the right. If anybody has any objections to that, please let us know.
Next waypoint.
Yeah, that sounds good to me, Cheryl. Does that mean we're at the top of the short slope that the short escarpment that we were investigating today?
That's what the nav has said to me.
Yeah, go ahead.
Yeah, I guess we should take advantage of whatever hard substrate we see.
Yeah. Yep, we're within like 40 meters of the waypoint.
Hi guys, this is Chris Kelly calling from Hawaii.
Hi Chris, how are you?
Good, I'm doing well. So I'm a little bit late. Sorry, I'm a little bit late to the party here, but looking at the sponges you were imaging just a few moments ago, and just a couple comments on it. That vase sponge, I'm not sure if somebody else ID'd it. It looked like it's in the subfamily Corbitellinae to me.
I like it.
And it's one of these sort of softer group of sponges, similar to some of the euplectellid vases. The big sort of yellowish ones, they look like they were demosponges to me. But again, I don't know if anybody else, I'm not on the chat room, and I'm just taking a quick look before I head off to work. But that was pretty interesting.
Do you want to try to line up on the trail?
Looking sponge, and I didn't see whether you got a photo of that by any chance. Did you get one of that particular sponge?
We have not made a collection yet, so would that be of interest that we should collect one of these?
Well, I think Chris was asking about a close-up.
A close-up.
040.
Yeah, we did get a
maybe if we
close-up
do like a
of polymastia.
045 or 050 just to get it
Yeah,
back
that's what
off
I
of it
thought
a
it
little
could
bit.
be, a polymastia, but I didn't, when I started watching, I hadn't seen a close-up of it. Yeah, I was just curious to see what it was.
We'll see if they want to collect anything.
Yeah, we've seen a few of them today.
Definitely a little bit of current pushing me straight back where I'm facing now.
Would you like another close-up of one, Chris?
If you want to move off it, I'd do it more.
Yeah, if one comes up, that's fine.
We could do
so these kind of yellow moundy ones, again, I don't think
we
they're
do 20 at 5.5.
I think they're demosponges. You did have sort of a close-up of one when you were imaging the Permissiid. But Yeah, I think any of these, I just don't know the Atlantic sponge fauna very well at all, so it's hard for me to sort of render an opinion as to whether they're collectible or not. That's a pretty interesting little community.
Chris, are you familiar at all with the genus Phakelia? That was one suggestion for what these are.
Which genus, excuse me?
Phakelia, Phakellia.
No, I'm not familiar with that, but I assume that's a demosponge. Is that right?
Yep, that is correct.
Yeah, then that would be consistent with what I was thinking. But that's great. Looks like you've got somebody who really knows the fauna there. Whoever ID'd it, did they give you an opinion as to whether or not it should be collected or not?
We haven't had any strong opinions on collection yet, and I think that was Rachel Barrett who identified that one. So we're not certain. We had Joanna on earlier. She's gone, and Shirley hasn't been able to get on today, but yeah, we certainly could use their help.
I guess my opinion would be that those yellow ones are pretty dominant, at least at this particular area.
Do a quick
might
video.
Worth
we can do lasers
getting
off.
a specimen of that because they're so numerous compared to the other ones. Looks like you have a Seriantid coming up too.
Looks like there's a little crab on the back of the sponge too, a hermit crab maybe?
Yeah, you can go tighter.
Well, it's a beautiful little scene.
So at the base of this glass sponge, are those hydroids perhaps, or
actually it's like an octocoral, maybe a...
Or is that an octocoral? Yeah, I don't know the difference. So I thought maybe we could be educated on that, because I often hear hydroids mentioned, but I don't know how to identify them.
I can't quite see them yet. I've changed the screen. Yeah, those are octocorals. They look like they must be stoloniferous octocorals, which the term means they grow like a ribbon, and you have individual polyps arising from a ribbon. And Leslie, I still don't have that great a view of them, so I could be wrong.
Okay.
But I think that's what they are. But the way to tell the difference between the hydroid and the octocoral, at least one way, would be to look at the tentacles themselves and see if the tentacles have small pinnules coming off them to make them look like feathers, in which case it would be an octocoral. If they didn't
We're going to move a little closer,
side branches,
get you a better view.
It's more likely to be a hydroid.
Thanks.
Now we're going to reposition a little bit to take a look at what might be another octocoral, a pink octocoral to the left of where we're facing now.
Just going back to what Chris Kelly said, that would be fine with me to collect a piece of one of these sort of larger plate sponges as a representative of what appears to be dominant here.
Okay, you can zoom
Yeah,
in.
I totally agree. There's enough of these to say that they're dominant. Would this little one be okay? It would definitely fit fairly well in the bio box compared to some of the bigger pieces, bigger sponges.
Well, this sponge actually looks like a glass sponge. It looks a bit different than the other one that you got a close-up on. It's just kind of a bit of a dirty glass sponge that's got a lot of sediment on it. But you could see some of the structure of it. So I think this is a little different than those other mounding ones. I didn't know there was this guy in here.
It's got some light for you.
I'm not absolutely positive what the idea is on this, but it's probably one of what they call sort of a brittle or a hard glass sponge.
Yep.
Okay, maybe we'll look for one of the bigger ones to be sure we're looking at the species that is the dominant one in this area.
Yeah, that's what I think, because this looks fairly different than that other one when you were imaging the coral a few minutes ago.
Okay.
It's a really nice close-up there, and so I do believe that those are octocoral polyps, and so I can see about 4 of them between the sponge and the feet of the urchin there, and I suspect they're all connected together. We just can't see the connection. Nice observation.
Cool. You can come out.
Ready for me to hit those?
Can anyone talk
you
about
can
what's
ask
going
video.
on underneath that urchin?
Video's clear.
Let me know when you're
He
ready.
seems to be
Then
grabbing
turn
at
this
some
back up.
organism down underneath him. Did anyone see that or notice
Video,
that?
let me know when you're ready.
I was wondering if there was a little bit of seaweed under there that had drifted to the bottom.
It
said
looked
go
like
ahead.
it had the...
Feeding on that.
Yeah, to me it looked like a piece of sargassum, a blade
It
of
looked like
sargassum.
sargassum, yeah.
Okay. So we're moving on. We're looking for one of the bigger plate-looking sponges. Is that right?
Oh, I see Shirley Pomponi's in the chat room now, so now we're in trouble with the sponges.
Okay.
No, actually, the cavalry just
That
arrived.
one over there? Okay.
Want me to turn to keep you in view?
Sure.
Okay.
What's up, Levi?
I'm trying to remember the name of that coral.
Is that bubble gum?
Yeah, but it's a...
Oh,
maybe it's not.
tip of the tongue.
Yeah. Pretty good delta. Does that trail keep going?
Thank you.
It's hard to tell.
This is looking a little bit like perhaps a Paragorgia bubblegum coral and maybe a black coral behind it, that orange one.
Nice.
Great. Yeah, I was thinking it looked like a Paragorgia.
All right. Zoom in video.
Paragorgia, it looks like it has tipped over at some point in the past and is now regrowing upwards.
Yeah. Oh, that's great.
Yeah, so this is definitely in the genus Paragorgia, the family Paragorgiidae. Maybe I shouldn't say definitely the genus Paragorgia. It's definitely the family Paragorgiidae. And it would either be Paragorgia or Cibogagorgia.
Right. Thank you, Scott. I wonder if Santiago Herrera is online, because he might be able to tell us the difference between those genera. Would this be one that we would want to make a collection of?
Okay, let's come out. We can look at the black coral while they're talking about sampling.
No one's jumping up and down wanting a collection of this, so we're going to take a look at the black coral behind it, which may be a black coral, at least from a distance. That's what it looks like.
Okay, go ahead, video.
Co-pilot, you want to fire the Niskin as a test?
Do we want to do that?
Yeah, we did one at 50 meters. Looks like no one's done one yet.
So this is definitely a black coral. Gets its name from the color of the skeleton, but you can see that the tissue can be a number of different colors, in this case kind of orangey brown. And this may be the genus Storopathes. Not 100% certain of that, but that's probably a pretty good guess. And you can see, like the octocorals, this is a colonial animal. It has a number of polyps, hundreds of polyps covering the colony. And each of the polyps is surrounded by 6 tentacles, and the mouth would be in the center. Now, this group of black corals is a little bit unusual because the polyps themselves are stretched. So if you look at any of the branches, the tentacles appear to be more in a line instead of in a circle.
Yeah.
Basically, you would count 3 pairs of tentacles, and then if you look in the center pair, you see there's a little raised nubbin. That's actually the mouth. And so those 3 pairs constitute one polyp. And all the polyps are connected to one another, so they basically essentially have a continuous gut.
Okay, come out.
Thank you. Gosh, that's a beautiful shot.
All right. Any decision on collecting, or should we move on? Okay.
The fan one.
Okay. So up here, would it be the one on, like the rounder one, or the one to the left of it? Okay.
Do you want lasers on it?
Sure.
Okay.
Yeah. So these are quite large.
That one out, or
We can do a portion of it. I don't think we'll be able to get the whole thing. Well, potentially.
put in the outer guys.
Yeah.
Stash it in there.
You can try.
Wonder how like firmly attached it is. I wonder if it's just sediment, you'll get the whole thing, you know, or if it'll pluck off or fall apart. Maybe just do the trim method. Cutters on the bottom. And
Yep.
yeah.
So we're setting up to try to make a collection here of the sponge. Is this the one that Scott was referring to as the dominant one in the area? And is this one alive? It looks very sedimented to me.
Standard 430.
Yeah, this is I think what Chris Kelly was saying. These sort of yellowish ones that are so common in this area. So yes,
Yeah,
is that
430.
sponge.
Okay,
50
great.
meters, yeah, 430.
The one above the lasers, not the one below the lasers.
Correct.
Oh, local off bottom, 1536.
Okay. If you're all studied up, Andy, you can
And we
change
have
it to mini Zeus.
another can,
Okay.
marine debris in the background of
Yours
the
spun up,
sponge.
But this hydraulic pressure says no power. Sometimes I'm seeing it, sometimes I'm not. So I think it might. Oh, there it goes. Now it is.
Okay.
So
I think
fine.
we tend to just go like 3/4 on it.
Okay.
It's just because it doesn't give you any increase in
Yeah.
pressure, and then it,
Unless using the craft, then I would use. I'd go 100%.
Which we are using the craft.
And based on what Chris said, I think that when the claw
Even
goes
flow.
for this,
It doesn't change
it
from the other,
may
does it?
break up
3/4
You can
somewhat.
make RPMs to tell you.
So pilots can do with that what they will.
It'll give you more flow, I think.
Yeah, okay.
Yeah.
It might make it fit into the
think-
bio box easier.
I think just as a, I mean, it doesn't matter, but it, prevents it from burning out the motor faster. with
Okay.
Slightly lower RPM.
Okay.
Just good practice.
Less wear and tear on the unit.
Yeah.
Okay. Understood.
Okay.
Yeah, you got your wide open, whatever box you want, according to my notes.
Pots look stable. Okay, hydraulics are up, and the arm is coming on. All right. So why don't you go ahead and tilt Mini Zeus down for me? And we're definitely going to go port bio box.
Okay.
Ooh.
A little thinner than I thought it would be.
Ready to come out?
Sure.
Okay. I'm hitting that sponge.
That's
So
okay.
Just go for it.